Thanks for the introduction. And yeah, I'm going to talk about the trajectory or path, how we identify the distinct subset of human hematopoietic stem cells that resist regenerative stress and thereby preserve stemness. But of course, first of all, we always show a hematopoietic hierarchy. But lately, it has been like there are a lot of these kind of hierarchy depictions out there that it made it difficult to choose from. What I want to highlight here is also we now appreciate there's lots going on down here. All of these hierarchy have in common that the HSCs are still on the top of them. But also lately has to be questioned whether the HSC, so the hematopoietic stem cells, are actually that important throughout native or steady state hematopoiesis, only about important about like really strong stresses like as transplantation. But clonal tracking studies in mouse then nicely elucidated that, yeah, once in a while, they can occasionally get activated and fulfill this pool of progenitors. And we now appreciate the progenitor pool as mainly taking care of the native or steady state hematopoiesis more, but still the HSCs, of course, are required long term. And this is, of course, all based on murine clonal tracking studies. In the human systems, we don't have this ease to look at these kind of longitudinal fates. And on top of that, in the human system, it has come also to recently be shown that they're in this kind of a cloud transcriptionally. So this cloud means it's like a continuum of low primed and undifferentiated cells according to single cell transcriptomics. So also we know that long-term HSCs and short-term HSCs, which are interchangeably used in the human system with multipotent progenitors, are in this cloud with other progenitors along a continuum and not really distinct, at least not transcriptionally or also by chromatin accessibility. But in this cloud, they have this unique function, and the function is determined by, of course, their differentiation capacity and their self-renewal, what we nicely always show with this arrow. But most of the time, they actually reside in quiescence. And this is not usually omitted in these graphics. So it's also clear that these programs are all opposing to one another and can't be executed all at the same time. So there has to be some kind of sequence or an order in a sequence or some kind of longitudinal integration of these fate choices. We actually got some pretty good insights lately from gene therapy trials. So the clonal tracking tool we have in the human context, because nowadays we have here up to 5-year follow-up in gene therapy trials due to the lentiviral integration, random integration. We can use that as a clonal tracking tool and thereby infer basically the fates individual clones undergo post-transplant. And what these studies showed was this kind of seemingly coordinated waves in a longitudinal fashion. And here it's implied by basically more functionally than really like phenotypically. So there's initially a wave of clones that respond and produce a hematopoietic output, but then disappear. And here short term means still like more than a year. And there are clones that remain latent, and they're only reading out or producing or contributing to the hematopoietic output later on. So what really like struck me was about this data was how are these clones here initially resisting this strong stressor of transplantation, but then later on produce progeny and read out? How are these fate decisions made? And we had previously a study on leukemia stem cell latency, where we identified the gene INCA1, barely studied gene, as a promoter of quiescence and self-renewal in leukemia stem cells, but it still permitted limited blast differentiation generation. Given now that leukemia stem cells are equally defined by these characteristics and that are shared, of course, with HSCs, we were wondering if INCA1 could also navigate between these fate choices in the normal context. But first of all, what is INCA1? INCA1 is, there are 5 publications with ours out there, and it's still in most data that's C3 of 54 or FAM2128. But what it's known, it's an inhibitor of the Rho-GDPase effector kinase PAC4. And we could link in our leukemia stem cell context INCA1 to a reduced levels of H4K16 acetylation. But we couldn't resolve if that it's the direct or bystander effect, because also this mark is very complex. It's not the standard acetylation relates to transcriptional activation. It can on a single gene basis, but not globally. It has also been shown in the hematopoietic context across a hierarchy here by epitope, so cytoph, single cell mass spec in the end, that different subpopulations have different levels of global H4K16 acetylation. Furthermore, a long time ago, it was already shown that H4K16 acetylation is fluctuating across the cell cycle. And we also see that in stem and progenitor enriched cord blood cells that the levels fluctuate heavily throughout the cell cycle. So yeah, to also see whether these two markers, so to say, play a role now in normal hematopoiesis, let's just directly zoom into the long-term HSCs. And what we observed there was pretty striking, given that, like, we know about the heterogeneity and the continuous in single cell transcription. But here we saw this clean-cut dichotomy between the single cells that are either Inca1 positive or H4K16 positive or double dim. And if we now convert these images, also these double dim cells, into binary images, you can appreciate that also in these double dim, we see intracellularly this mutual exclusivity of these two markers, hinting at that there might be a direct regulation of them. But so these cells, these Inca1 positive cells, are roughly 10% in the long-term HSC compartment, but we also find them in the other three intermediate and short-term HSCs, as you can see here. In follow-up studies, however, we only saw in long-term HSC that the Inca1 high cells coincided with low CDK6 levels, and not in the short-term HSC. So it's highly context-specific, but still these cells telling us that we have potentially alternative states of quiescence within the long-term HSC population. Because CDK6 is a marker of early HSC priming out of quiescence, or primed HSCs for quiescence exit. And this places now the Inca1 high cells in the long-term HSCs at the deeper end of the quiescence spectrum. This was further supported then also by single-cell RNA-seq data that we generated from mobilized peripheral blood, so as a source of in vivo activation. And using RNA velocity and diffusion pseudotime algorithms, we were then able to infer this activation trajectory that ordered the single-cell transcriptomes along the diffusion pseudotime. And this was following also the activation could be seen by CDK6 level increasing with the inferred pseudotime. And we also saw then an increase for PAC4, whereas Inca1 high expressing single-cell transcriptomes were enriched early in the diffusion pseudotime. Of course, we can't look at H4K16 acetylation dynamics in an RNA context, so we opted for multi-time point confocal microscopy and harvested at multiple time points the cells during the very first cell cycle out of quiescence, and they were and thereby generated single cell data on 3,500 cells, and now face the problem that we saw a lot of heterogeneity, but this heterogeneity was now due to the cells not being equally synchronized. And as you can appreciate here, we can still see that these markers all increased throughout the cell cycle and a little bit faster in short term than in long term. But we couldn't really discern whether this cell is this cell or this cell is this cell and so on, and how these markers relate to one another. So again, we opted for diffusion pseudotime and to rather order the single cells according to their confocal parameters than the actual time point harvested. And as you can appreciate here, the diffusion pseudotime also ordered roughly the time points harvested according to in a longitudinal fashion as you would anticipate. And also, as expected, the progenitors progressed faster through this pseudotime and the long-term HSCs lagged behind. But this analysis now allowed us also to, by regression analysis, to infer the sequence of events. So by that, we saw that H4K16 acetylation and PAC4 get actually upregulated even before CDK6 protein upregulation occurs. So this places now these two markers early in the quiescence activation and also suggesting that Inca1 might impede this activation. To now look further into that, we overexpressed Inca1 in long-term HSCs. And as you can appreciate here, Inca1 suppressed all three markers, prevented the upregulation of these three markers in long-term HSCs, but it didn't do so in short-term HSCs. So they were not permissive to Inca1 overexpressing similarly. We also saw a similar pattern then in RNA-seq data generated from these cells that also in long-term HSC and short-term HSC, Inca1 expression enriched for common stem cell signatures. Particularly in long-term HSC, we saw suppression of signatures associated with stem cell activation, here from the Murren study, or in the hallmark genes that generally a lot of signaling and immune responses associated with HSC activation were downregulated, and we had this global resistance or this global downregulation of the hallmark gene sets. So is this linked now to H4K6 denacetylation or the dichotomy we observed initially? To answer that, we went literally on a fishing expedition and fused Inca1 and PAC4 to BA N and C terminal. And BA is biotinylating proteins in close proximity that then can be identified by mass spectrometry. Thereby, we could generate this Inca1 PAC4 protein interactome. And I overlaid here the expression of these individual interactors from the long-term HSC RNA-seq data set. And just to show that this interactome is highly expressed in our context. Strikingly, we found the direct link between Inca1 and H4K6 denacetylation, with SIRT1 being an Inca1 interactor. SIRT1 is the major H4K6 deacetylase. We also confirmed this interaction then in our context by Inca1 by proximity delegation assays, showing that a signal for Inca1 and SIRT1 in HSPC, so in the stem and progenitor enriched fraction, roughly at the same frequency as we would expect from the Inca1 staining previously. And we didn't see interaction really in the progenitor cells. We also saw that Inca1 sequesters or recruits SIRT1 into the nucleus when overexpressed. And that this interaction also occurs at the chromatin. We used chromatin fiber assays on HSPCs, where we see that in the chromatin fibers, Inca1 is coinciding with SIRT1, whereas SIRT1 is mutually exclusive to H4K6 denacetylation. But what really caught my attention in this interactome was PVL2, or CD112. CD112 is a cell adhesion molecule, and it's connected via aphadine MLLT4 to the cell's side to actin. The thing about here PVL2 is that it was the only protein we extracted with all 4 constructs, and aphadine was the strongest or most abundantly retrieved interactor for Inca1. So how does CD112 look in the haematopoietic context? It basically looks very similar to CD34 surface expression, and in the HSC compartment, it's slightly higher on long-term HSCs and short-term HSCs. When we did then also immunostainings and to confirm that we have Inca1 PAC4 interaction or like close proximity to CD112 on the surface, and indeed high density regions coincided. But more strikingly in this data was then that we saw that Inca1 high cells were CD112 low and vice versa, and that's also shown here in the 2D plot. This was also further confirmed that by Inca1 overexpression in cultured long-term HSC, that it reduced the levels of CD112 surface levels. But intracellular staining actually showed us once in a while that these are few cells upon Inca1 overexpression, where we saw this strong intracellular staining of CD112. And so could Inca1 be responsible for its uptake And so we did a staining with LAMP1, an endosomal, late endosomal or lysosomal marker. And indeed, we could see that LAMP1, INCA1, and CD112 coincided here in these pointed structures. So could CD112 actually allow us to prospectively isolate INCA1 high cells and H4K16 acetylation high cells? To answer this, we sorted long-term HSCs on the very ends of them, the 20% edges, the extremes, for CD112 high and low, and used all the short-term HSCs as a control. Thereby, we could establish that in the high cells, in the CD112 high cells, we see the highest H4K16 acetylation and the least INCA1 expressing cells. And in the low population, we saw the lowest CDK6 levels and the lowest ROS levels as well, placing them at the deeper end of the quiescent spectrum. We also conducted RNA-seq on these populations, and were really surprised that despite them being so close together, we got roughly close to 300 differentially expressed genes. And among the, one of the pathways upregulated was mTOR signaling, which upregulated in the more prime population. That's very reminiscent of muscle stem cell G-alert phase. That's also quiescent, but already like poised the cells to respond to regeneration. Among the 5 parts that were upregulated in the prime fraction, 4 of them were also suppressed. We saw suppressed in our INCA1 overexpression data set. And again, showing us this inverse relationship between CD112 and INCA1. Furthermore, the biological processes are very nicely aligned with actually all this mechanism we just talked about. So cell adhesion, actin, and GDPase signaling and so forth are enriched in the CD112 low fraction. So telling us again that we might have potential linked then in the immunophenotype with these states, and INCA1 at the center coordinating this surface exposure of CD112, but simultaneously also the chromatin state in the nucleus. And now it's of course a question, do these states also translate into stem cell function? For that, we overexpress now INCA1 in our xenotransplantation model. And we use BFP, this co-expressed with this vector, to track relative engraftment compared to the overall engraftment that usually steadily increases until a plateau is reached. And if it's a neutral engraftment pattern, you would expect something like this here by the control inferred from this data set, here expressed as a relative ratio to input. And we saw the same for INCA1 at 2 weeks and 20 weeks. However, at 4 weeks, we saw this 4-fold reduction, so this is a log2 ratio, relative reduction to the input, that basically tells us that the BFP positive fraction expanded less than the BFP negative fraction, and we interpreted that as a delayed repopulation kinetics. So meaning that we have here actually quantitatively less cells, but what about their quality? So we used from all 3, harvested from all 3 time points the human cells and transplanted them into secondary animals. And in all the INCA1 overexpressing cohorts, we observed that more mice engrafted. And at 20 weeks, we had enough cells to conduct limiting dilution assays and could determine that also the engraftment levels at 20 weeks were basically the same. The INCA1 overexpressing cells had 4.5-fold higher stem cell frequency. Telling us this early restraint might translate into higher preserved stem cell capacity. We also then wanted to confirm that whether PAC4 inhibition in a different setting, so PAC4 knockdown, could recapitulate this longitudinal phenotype. And what we saw was that we saw the same transient reduction with 2 different PAC4 constructs, and we could also see that that translated then into higher secondary repopulating capacity. So we were wondering what's happening here at the 4-week time point. Are these cells in quiescence just not reading out? What if we give them another stimulus? So we did the same experiment again, and this time applied a 5FU stimulus to deplete basically the proliferating cells, testing whether these cells are quiescent or if they respond to demand generated. And while we did not really see a lot in the primary mice, upon secondary transplantation, the control cells did not engraft at all, the control with 5FU stimulus, where the Inca1 overexpressing cells with 5FU stimulus even showed enhanced self-renewal capacity, higher stem cell frequency. So obviously the stimulus activated the cells to self-renew. But what about now if we do this longitudinal analysis without culture, without gene modification, and use prospectively isolated CD112 low and high long-term HSCs? We used 3 independent cord blood pools and saw in all of them high variability. But what was consistent was that the CD112 high population that is more primed, according to our in vitro data, showed this early, more rapid response in a population, but then declined, whereas the CD112 population lagged behind. But this delay also, again, translated into higher stem cell frequency, again, implying that delayed repopulation preserves stemness. So if we take this now together, we have now a little bit of higher resolution in the stem cell compartment, where we see alternative states of quiescence that upon transplantation basically predetermine the fate of these cells, either to immediately contribute to the hematopoietic output or to remain in latency, resist the activation, eventually produce progeny, but still maintain self-renewal potential. And in other words, with latency integrates basically the 3 hallmarks of stemness. We can also look at this a little bit different as like a reservoir, and that is defined by the markers we discussed in quiescence, where we have a latent long-term HSC reservoir that is not called upon as long as progenitors are there, but late in after transplantation or secondary transplantation, or if we deplete this pool by 5FU, they are recruited, can self-renew, and basically then generate the hematopoietic output. Interestingly now, interesting for the future would be, of course, now if we can harness somehow these insights in the population dynamics, but also in the mechanistics to maybe awaken latent HSC to combat age-related immunodeficiencies at old age? Or can we extend latency, suppress mutated HSCs for contributing to clonal hematopoiesis? And with this, I'd like to thank everybody involved in this project, the whole DIG Lab, our collaborators, our funding, and you for your attention. Thank you.